Okay, so I'm going to talk to you about an experiment that I carried out actually during my PhD work, which was at University College Cork. And before I begin, I'd just like to talk about a pattern that's been observed from a number of different systems which formed the basis of the study that I'm going to present here. So to Chile then, and Juan Pablo Castilla's work on two marine reserves, Las Cruces and Montmar. And he found that after designation of the marine reserve, due to the cessation of fishing and other human activities, that there was an increase in the density of a predatory gastropod, a subsequent decrease in the density of a bivalve, which was a dominant competitor for space in the rocky intertidal, and this led to an increase in the diversity of several algal species in the marine reserves. And then Pablo Marquette and his colleagues went on to look at certain patterns as a result of these cascading effects. And Pablo and his co-workers compared the body mass abundance relationships inside the marine reserves to areas of coastline very nearby, and found that the slope of these body mass abundance relationships was maintained in spite of these cascading effects. And a similar pattern has been shown for freshwater lake in Michigan, USA, Tuesday Lake, where in 1984, 3 species of planktivorous fish were removed and replaced with 1 piscivorous fish, which also led to cascading effects in the lake 2 years later. So Thomas Janssen and Joel Cohen and some other colleagues show that this change in the fish community led to a significant increase in the biomass abundance of secondary producing zooplankton, which led to a subsequent decrease in the biomass abundance of phytoplankton and thus primary productivity in the system. But again, in spite of these cascading effects, they show that the body mass abundance, or the slope of the body mass abundance relationship, was conserved between these 2 years. So you might ask, why is this important? Why should these body mass abundance relationships matter? And people like Damath and a whole range of other famous authors such as James Brown and Engquist have shown that body mass is thought to scale at a global scale to the -3/4 power with abundance. And body mass is also believed to scale with metabolic rate to the +3/4 power. And Damath, in a classic 1981 Nature paper, combined these two relationships to produce the energy equivalence hypothesis, which suggests that ecological and physiological processes somehow combine to mean that resources are divided equally across all species populations, irrespective of their body size. So my question then was, does this pattern hold for other aquatic systems? And if so, can we identify the mechanism driving this pattern? And my study system, which Peter has already talked a bit about in his talk this morning, was Lough Eoin in southwest Ireland, a beautiful spot. Also designated Europe's first marine reserve in 1981, and it's been studied extensively since the 1920s. I promise I didn't steal this slide from Peter, but that is again Jack Kitching in his fancy Scuba Pro diving gear at Lough Eoin in the 1930s. And taking an existing experiment, which I carried out during my PhD, I had found, I don't want you to focus too much on the colored lines and all the patterns in these figures here, but basically in the left-hand side, I found cascading effects on ecosystem functioning as a result of some manipulations I carried out to cage mesocosm ecosystems during my PhD. I found increases in secondary production, decreases in primary production, and I also found effects on the stability of the food webs within my cage system. So I thought this would be an excellent opportunity to ask, what happened to the body mass abundance relationships within my experiment? So I'll get a little bit more into detail of what that experiment was now. And it was a very large caging experiment, which I carried out over about a 16-month period. And I chose 10 prominent benthic consumers from the Lough Eoin system, and I was intent upon manipulating their diversity as a top trophic level within these cage systems. So I placed these consumers into my large subtidal cages, sealed them off so that they couldn't escape, and I placed them in the shallow subtidal, where they stayed for a period of 16 months. And over that time, because of the small mesh size of the cage, small invertebrates such as amphipods, isopods, bivalves, gastropods, polychaetes could recruit naturally into the cages and form complex communities around these manipulated consumers. So just to give a little bit of information about the experimental design then, I had an initial community assembly phase which took place over 6 months. I had 24 replicate cages, and I placed one individual of each of these 10 manipulated consumers into the cages, matching them for body size as much as I could to try and create very similar initial communities. And then I allowed the cages, put them in the shallow subtidal and allowed natural communities to assemble around them over the course of that 6 months. So what I was trying to do was create replicate communities, if you like, and I sampled them a few times over that period and monitored different properties of the system, measures of ecosystem functioning, diversity, and trying to describe the food web within the cages as well. And based on some other experiments where I had calculated or estimated the interaction strength, the average interaction strength within the system of those 10 consumers, I identified who were the the strongest and weakest interactors out of those 10 manipulated consumers. And that then became the focus of my manipulations after this 6-month community assembly phase. So I had 6 treatments with 4 replicates of each. So one set of 4 cages continued as before with the manipulated consumers intact, the same 10 manipulated consumers. I had another set of 4 cages where I removed the 2 strongest interactors, another 4 I removed the 3 strongest interactors, more where I removed the 2 weakest, the 3 weakest interactors, and finally so the 3 strongest and 3 weakest interactors removed. And I arranged the treatments in a randomized block design in the shallow subtidal to try and minimize the natural variability in the experiment. So again, I would have sampled it throughout the remainder of the experiment, which was another, 8 or 10 months, and I measured at each sampling session the species that were present within each of my cage mesocosms. Using a combination of gut contents analysis and literature research, I measured the interactions between the species in that system to compile food webs like you see in the picture here, and this is a food web from one of the cages, so these were very complex communities, 50 to 100 species in every cage and thousands of interactions. I also measured the density of every species within the mesocosm at each sampling session, and also the body mass of every individual identified in the experiment, which is quite a sizable number. And then to address this question about the body mass abundance relationship and how it might differ between the different treatments, I chose one sampling session before the interaction strength manipulation at this community assembly phase, where I would have assumed that all the communities were operating at a similar level. They were at that stage replica communities, if you like. And then I choose one sampling session after the interaction strength manipulation. Ask the question, does a trophic cascade occur between these 2 sampling sessions? And if so, do these cascading effects alter the body mass abundance relationship? And I also examined both average species body mass to look at local size density relationships, and also the body mass of individual organisms to look at size spectra or individual size distributions within the system. So to the results then, and first of all, to that question, is there a trophic cascade? The answer was yes. So if we look at the graph here, the white bars represent that sampling session before the interaction strength manipulation, and I found no significant difference in the secondary production, which was measured as the log 10 biomass density of grazers and detritivores within the system. So there's similar levels of secondary production across all the treatments at that stage. The grey bars then are the sampling session after the interaction strength manipulations had taken place. And I saw that removal of the 2 and 3 strongest interactors led to a significant increase in secondary production. And also the removal of 2 weak interactors led to a significant increase. This then had a cascading effect on primary production, measured as the square root of chlorophyll A within the system on glass slides. And I found that the removal of 2 and 3 strong interactors led to a significant decrease in primary production. The question then, what effect did this have on the size-density relationship? There's a lot of information to take in here. I'm not expecting you to understand every single one of these graphs, their comparisons before and after the manipulation, but maybe just to sum it up, basically what I've done is I've plotted the log 10 of body mass against the log 10 of the abundance, the average body mass of every species within the system and the average abundance of every species within the system to look at these size-density relationships. Because these communities are local communities fed by the wider species pool from the Loch-Eoin system, they are local size-density relationships which differ from global size-density relationships, and they tend to be very variable. So the widespread that you see in these plots is not unusual for local size-density relationships. I'm going to drown you in a sea of numbers. So this is basically getting at the nitty-gritty of what are the differences in the body mass abundance relationships between the different cages. And what we want to focus in on is the comparison, first of all, the intercept of these relationships and how did that change when I compared the treatments before versus after the interaction strength manipulation, and the answer was every treatment was significantly different. However, there's a strong suggestion that this is a seasonal effect, because if we take just the sampling session after the interaction strength manipulation and compare the intact communities to each of the other treatments where species were removed in turn, we see that there was no significant difference between those intact, the intercept of the body mass abundance relationship in the intact communities versus the treatments where species were removed. Interesting enough that when we look at the slope, we find no significant difference before versus after the interaction strength manipulation, and also no significant difference between the treatments where species were removed compared to the intact community. And we might ask then, well, what are the consequences of this for, say, like the energy equivalence hypothesis? Well, first of all, I mentioned that my communities were local communities, and I said earlier that the slope of global size density relationships were predicted to be -3/4. The slope of local size density relationships are thought to be much closer to -1/4 scaling. A study by Tim Blackburn and Kevin Gasson in 1997, where they looked at hundreds of different communities and measured body mass abundance relationships. The average slope of my communities was very close to this -1/4 scaling, which suggests that the energy equivalence hypothesis does not, well, it wouldn't be supported within my systems, and in fact, large-bodied species should consume less energy per unit biomass than small-bodied species. And this is a possible explanation for a trend that we observed throughout these studies at Loch Eoin, where smaller individuals were found to have stronger per unit biomass trophic interactions than larger species. But one problem with those relationships I showed you, they have very high variability, and you could argue that drawing a line through those data points anywhere, because of the large standard error associated with, you're probably not going to find a difference in the slope or of those relationships. So we wanted to find something that would be a tighter fitting, something that might give us a little bit more confidence that the patterns that we observe are in fact accurate. And how we thought to do this was to look at the size spectra within the cage systems. So these are much tighter fitting with R-squared values of 0.8 or higher. And what we've done is to assign individuals to body size classes and plot the log 10 of the midpoint of those body size classes against the log 10 of the number of individuals per body size class. And again, fit a body mass abundance relationship to this. And in the same way as before, another sea of numbers, but this time we find very similar patterns to the local size density relationship. So again, if we look at the comparison of before versus after the interaction strength manipulation, we see significant differences across the board. Again, the suggestion this is a seasonal effect where if we compare the intact communities to the cages where species were removed, no significant difference in this intercept of the body mass abundance relationships of these size spectra. Again, no significant differences in the slope of these size spectra before versus after the interaction strength manipulation, or when we compare the intact community to the cages where species were removed. And so you might ask then how, the key question, how is this maintained, this body mass abundance relationship? And as certain species increase in body size and abundance as a result of the trophic cascade that I described earlier, they create, in effect, gaps in the size spectra, which are then filled by potentially maybe new species coming in to optimize this niche where they can get an energy requirement that would most be suited to their body size. And if that is the case, then we might expect to see an increase in the species turnover within the system if indeed new species are coming in to replace or in fact fill these gaps in the body size spectra. And this was the case. So when we looked at species turnover, we found that the lowest species turnover was found to be in the intact communities, and there was a significant increase in species turnover after the removal of 2 and also 3 strong interactors, and also 3 weak interactors. So this suggests that this could be, in fact, be a primary mechanism for maintaining the body mass abundance relationship. So just to sum up then, we see the body mass abundance relationships are indeed robust to cascading effects. This is true whether we consider average body mass or the individual organism size spectra. We found species turnover to be one of the primary mechanisms that is underlying this effect, and this should have implications as a stabilizing process then that would maintain the distribution of energy and nutrient cycling across species populations. So just to acknowledge University College Cork, my funding bodies, National Parks and Wildlife Service, and a whole load of field helpers as well, and thank you for listening.